Good morning, and let's see JPSC JET question number 57. Question is, in muscular dystrophy, the actin binding protein dystrophin is absent or defective. Dystrophin has similar actin binding domain and structure to the spectrin and alpha actinin. Which of the following is most likely to occur as a result of this deficiency? Options are enhanced smooth muscle contractility. Option number B is deficiency in skeletal muscle. Option number C is loss of binding of the I and M bands to the cell membrane. And option number D is loss of organelle and vesicle transport throughout the muscle and loss of integrity of the desmosomal component of the intercalated disc of cardiac muscle. So what we're gonna do is, this is the question. We will see about this question is related to cell-cell interaction and cell ECM interaction, isn't it? So we will see a video and then we will see what dystrophin is, what spectrin is, what alpha actinin is. These are component in cell-cell interaction and cell ECM interaction. We need to know about how these cytoskeletal structures are connected to each other. You know cytoskeletal structure are which give supports to the cell, isn't it? That is similar to bones that support the body. We will also see desmosomal component. Okay, so let's see a video and then we shall continue on this question. So you can see that two cells are connecting with some kind of fibers like structure, isn't it? So these fiber like structure are laminin and integrin connecting with cell-cell communication, cell-cell interaction. These you can see lipid raft on the plasma membrane. Inside the cell membrane, you can see the cytoskeletal structure giving support to the cell membrane. You can see the whole cell is given supported by the cytoskeletal structure, isn't it? The microtubule and the microfilament and intermediate filament. You can see the actin molecules forming as a cytoskeletal structure inside the cell. These are microtubules forming rod-like structure inside the cell. So very important, this vesicle is being transported on the microtubule from one place to another, say from endoplasmic reticulum to Golgi apparatus. And all the roads start from microtubule, originate from centriole, isn't it? Everything starts from centriole. The centriole is the center of the cell. So now that you have seen this video, and let's see what dystrophin, spectrin, and alpha-actinin is. So dystrophin is the molecule which is connected to actin associated cytoskeleton, as you have just seen, and you can see in the figure that dystrophin is connected to the cytoskeletal, isn't it? Again, in the next figure you can see dystrophin is connected to actin cytoskeleton. Again, in this figure dystrophin is connected to actin filament, which act as a molecular shock absorber for the cell, isn't it? Talking about spectrin is a bed like scaffold interconnected. Scaffold is an interconnected net like structure which gives support to the cell, you can see here. And alpha actinin is the protein that binds two of the actin filament to each other. If you remember the sarcomere and the myosin and actin protein discussed just in previous question, then you can remember that actin slides over the myosin slides over the actin filament, isn't it? Or and this is the molecular mechanism and sliding theory for muscle contraction. So these two actin are connected with the help of alpha actinin, isn't it? Here you can again see alpha actinin present. Alpha actinin, again in this figure you can see clearly two of the actin are connected with alpha actinin, and you can see how dystrophin is connected to uh, actin filament. Okay, so you can see in this figure that dystrophin is a complex of glycoprotein, isn't it? It is not one single protein. So now that you have become clear that what dystrophin is, what is spectrum, let us again come to the question. So the question was, in muscular dystrophy, the actin binding protein dystrophin is absent or defective. Dystrophin has similar actin binding domain and a structure to the spectrin and alpha actinin. Which of the following is most likely to occur as a result of this deficiency? So option number A is smooth muscle contractibility. So if the dystrophin is deficient, is not present, so how can this lead to enhanced contractibility? It will lead to muscle weakness and degeneration, not enhanced contractibility, isn't it? So the option number A is wrong. Option number B is deficiency in skeletal muscle actin synthesis. Dystrophin affects the anchoring of actin, isn't it? But is not associated with the synthesis of actin. Actin production remains normal, but it cannot be properly stabilized at the membrane because the connectivity is lost because due to the absence of dystrophin protein. Option number C is loss of binding of the I and M band to the cell membrane. So here you can see in this figure that dystrophin is connected to the I band, isn't it? I band or A band, isn't it? You can see here. So this is the correct answer. Dystrophin's role is vital protein that acts as a mechanical link. You can read the literature. I have told you what the concept is and from where the question has been asked. So dystrophin is connected to I and A band. So loss of binding to the I and M band to the cell membrane is the correct answer. And option number D is organelle and vesicle transport rely primarily on microtubule and motor protein, isn't it? So option number D was loss of organelle and vesicle transport throughout the muscle cell. So vesicle and membrane, organelle and vesicle transport, you have just seen that this you have just seen in the video that it was responsible. The vesicle transport is responsible by kinesin and dynein protein which travels on microtubule, and it has nothing to do with dystrophin, isn't it? So you have just seen in this video that we have seen. So option number D is also not correct, not the dystrophin. And lastly, uh, and loss of integrity of the desmosomal component of the intercalated disc of the cardiac muscle. So you know cardiac muscle, and you can just remember what desmosomal component is. Desmosome is a protein that connects two of the cell to cell adhesion molecule, isn't it? So you can just remember this cell adhesion molecule contains desmosomes, and various proteins like desmoplakin and uh, desmo desmoplakin and placoglobin like molecules are responsible, and desmocolin or cadherin, which we have seen in the previous questions. So that type of protein are responsible for desmosome integrity, isn't it? So dystrophin has nothing to do with this also. So that was question number 57. Thank you for your time.